In the last part of this week, we're going to talk just a little bit about eukaryotic gene regulation. The reason why we're going to focus on eukaryotic gene regulation is because they have many more ways that they can actually regulate how genes are being expressed and how much of those genes are being expressed. In eukaryotes, there are 5 primary levels or ways that they can control this gene expression. And today we're just going to talk about the first 3, the chromatin structure, transcriptional control, and post-transcriptional control. Next week we'll continue and talk about the translational and post-translational forms of control. So to go back to our chromatin structure, think about the organization of chromatin. If something is euchromatin, it's going to be genetically active. If it is heterochromatin, it is going to be tightly wound up and inaccessible for that RNA polymerase. And so genes are not going to be expressed in that particular cell or that particular region of the genome. Also remember bar bodies. These are going to be that inactive X chromosome in females. This chromosome is not going to be producing gene products, or at least very limited number of gene products. And so this is just one form of control that these eukaryotic cells can have. And you can just relate them to the degree that they have this supercoiling of their DNA, and whether or not this DNA is going to be accessible to that RNA polymerase or not. So if we look back, some of these levels of chromatin structure, you can see here are the histones. DNA is going to be wound around these histones. It's going to be in this octet. We're going to call it a nucleosome. It's going to be very tightly packaged up. The RNA polymerase physically can't get in there. Okay, and we talked about X inactivation. This is going to be where, through random assortment or random events, one of the 2 X chromosomes is going to be inactivated. Those genes are going to be, again, very inaccessible to the RNA polymerase as well as the rest of the transcription machinery. Okay, so talking about transcriptional control, this is probably, or arguably, the best studied form of gene regulation. And it's just because it's physically the easiest to study. So many people have studied different transcription factors. And these transcription factors are simply proteins that happen to bind DNA. And we call them transcription factors. And so what combination that you have, or that an individual might have, is believed to regulate activity of that particular gene. Enhancers are going to be regions where factors that regulate transcription can also bind. Silencers, another region where factors that regulate transcription can bind. Many of these are going to be already present in the cell. Many of them may have to be activated before they will actually bind to the DNA. So an example for you is cortisol, which is something I worked on as a postdoc, and this is just a stress hormone. What happens when levels of this hormone increase within the body is that this hormone will bind the receptor. This is on the cell surface, called the glucocorticoid receptor. When hormone binds, the receptor is going to change its conformation. It will go to the nucleus and bind the DNA. And it can then affect gene transcription by binding that DNA. And in the absence of hormone or lower levels of this hormone, the receptor usually does not bind DNA, usually does not get into the nucleus. So only upon higher levels of the stress does this happen. Okay, so another form of control is the post-transcriptional control. And what happens here is it's going to control the gene after the mRNA transcript has been made. So a couple of different things or ways that this can be regulated. First of all, excision of the introns can vary. Splicing of the exons can vary. Okay, this can determine the type of mature transcript that leaves the nucleus. It also can control the speed of mRNA transport from the nucleus to the cytoplasm. So if you have something affecting, say, the caps, affecting the tail formation, this RNA may not be able to leave the nucleus, and so you will never get translation from that particular gene product. So looking at some of the different combinations you can get, imagine here you have a transcript with A, B, C, D, E as exons, and then brown here would be the introns. In one cell, you may have splicing such that it keeps all of these exons, A, B, C, D, E, in the mature mRNA, which would give you this particular protein product. In a different cell, or under different conditions, the cell may take different exons, maybe only A, B, D, E, or maybe A, B, C, E, or maybe just A and B. Okay? Any combination of those it can take, and it can make this a completely different protein product. So you can see, as the number of exons increases, the potential complexity increases exponentially. So this week, I want you to focus on the Mastering Genetics homework for both Chapter 13 as well as go ahead and start Chapter 14. As always, focus on the discussion forums. This week, we will not have a quiz. However, next week we will have a quiz that's going to cover both Chapters 13 and 14. In your spare time, please also continue to work on your student project. So next week, we're going to focus on translation as well as further regulation of gene expression.